My name is Nancy Holbrook, and my research focuses on fungal endophytes that are found within Asclepias plants, or milkweeds, a plant essential to monarch butterflies. Before diving in, it is important to understand what a fungal endophyte is. Fungal endophytes are fungi which live inside plant tissues without causing obvious outward signs of disease. Fungi can live in leaves, roots, petals, stems, and bark, but in this project I focused on fungi within leaves. Why should we care about fungal endophytes found in milkweeds? Milkweeds are the main food source for the monarch butterfly. In addition, milkweed plants are important in serving as sites for female monarchs to lay their eggs. Once eggs hatch, monarch caterpillars consume the milkweed in preparing for their transformation to butterflies. What's so special about milkweeds? Milkweeds are a very diverse plant genus with over 50 species known for the latex fluid that they contain. Within this latex are chemicals called cardenolides. Cardenolides are toxic to most animals but beneficial to the monarch butterfly by aiding in its resistance to a parasite called OE. Different species of milkweeds have very different concentrations of cardenolides. Considering this chemical variation, we expected that maybe the fungi within milkweeds is also different across species. So, what questions are we asking? Do different species of milkweed exhibit varying prevalence rates of fungal endophyte infection? And do different species of milkweed harbor different communities of fungal endophytes? How did we examine endophytes among our 4 species of milkweed? We collected plants, picked leaves, cut leaf squares, and surface sterilized the leaf squares to make sure we were only growing fungus from inside the leaf. Then, we plated the leaf pieces on agar. So, what did we find? Interestingly, there was suggestion that prevalence of fungal endophyte infection was different among the 4 milkweed species, with high prevalence of infection in tuberosa and markedly lower prevalence of infection in curasavica. What about the communities of fungi isolated from the 4 species? We also found that the constituents of the fungal endophyte communities within the 4 species were different. In the figure, each color represents one type of fungal endophyte, and the size of the bars indicate the proportion of endophytes in that milkweed species that were a particular type. What can we conclude from these results? Different species of milkweed do seem to have different prevalence of fungal endophyte infection. In addition, different species also seem to harbor differing fungal endophyte communities. So, what are our next directions? We would like to replicate our study with a greater number of plants and sequence fungus that we isolated. In the future, we could possibly supplement monarch diet with the fungi isolated, beginning to test whether any fungi could confer disease resistance. Thanks for watching, and I hope you enjoyed this presentation.
Created using Powtoon.